In this lecture, we'll talk about transgenic fish. Transgenic fish is an organism that has a foreign or modified gene transferred to its genome using the in vitro genetic techniques. It is also called genetically modified organism (GMO) or a transgenic organism. Scientists are seeking ways to genetically engineer fish and other seafood species to introduce or amplify economically valuable traits. Fish are of particular interest to researchers since many fish produce large quantities of eggs, so those eggs being external to animals as opposed to mammals that produce fewer eggs internally makes it relatively simple to insert novel DNA. So there are several applications for transgenic fish. Research on transgenic fish is currently under development for at least 35 species of fish worldwide, as well as for a variety of mollusks, crustacean, plants, and marine organisms for various purposes. Transgenic technology has been successfully used to develop fast-growing superfish stocks for human consumption, and it can be also used to produce pharmaceuticals. To test water contamination in both developed and developing countries, several labs now have GM fish with increased growth performance caused by extra copies of growth hormone genes. So important thing is to select a selection of the species and what genes to be introduced in fishes. For the aquaculture importance, Indian major carps, common carp, channel catfish, Chinese carp, salmon, trout, and tilapia are the best species for the transgenic project. Improvement of growth rate. Imp, imp, so these some of these traits like improvement of growth rate, imparting disease, and environmental stress resistance are some important traits for the transgenesis. Gene transfer have been successful in several fishes. For example, common carp, rainbow trout, Atlantic salmon, and catfish. Salmon or rainbow trout growth hormone, chicken delta crystalline protein, winter flounder antifreeze protein, E. coli beta galactosidase, and E. coli hygromycin genes. So these are some of the genes which have been introduced into fish. So there is the advantage of creating transgenic fish. Fish have large and transparent eggs, allowing it to be easy for gene transfer. Most fish eggs are injected within an hour after fertilization because they are released from the female, and the first division of the eggs occur 1 hour after the fertilization. Salmonoid eggs are more difficult to inject because they have a hard outer covering called chorion. Survival rates for microinjected fish embryos are much higher, 35 to 80% than mammals. So it's very easy as compared to the mammals. So since the survival rate for microinjected fish embryos are very high, 35 to 80%, so 10 to 70% of fish being can be transgenic. So this is just this diagram shows how one can create a new variety of fish or a transgenic fish. In the first step, you can select the gene of the interest and put it in a vector, with a suitable vector which can be easily expressed in fish. So like in this case, like the promoter gene is taken from ocean pot fish, and the growth hormone gene is taken from the salmon. Growth hormone gene is from salmon, and the promoter is taken from the ocean pot fish. So this growth hormone gene from salmon is under control of ocean pot fish, and then you have it in a suitable expression vector. So the constructs are made, and then these are into these genes, these constructs are then linearized and then injected into the salmon eggs. So here these are the eggs easily, and then we can have the transgenic fish. So this is the easier way of developing transgenic fish. So there are several methods which can be employed to transfer genes in fish eggs. Microinjection is one commonly used. One can use electroporation, gene gun injection, use of retroviral vector also. Some vector which can infect. fishes, they can be used. Liposuction is another one. Use of embryonic stem cells can be used. Tissue injection, gene targeting. So there are several methods which can be employed to introduce gene into fish eggs. So some of these techniques like microinjection. So this is microinjection is the most common method of gene transfer in fishes. So soon after fertilization, gene is microinjected into the cytoplasm since the X nucleus is not visible in the fishes. So gene introduces cytoplasm. Lineage DNA rather than circular DNA is injected for greater probability of the former to get integrated into the host genome. So the vector actually is linearized first. So it has more chances of getting integrated in the fish genome. Higher amount of DNA is used for cytoplasmic gene transfer than when it is injected to the pronucleus, since the DNA will be introduced in cytoplasm instead of nucleus. So higher amount of DNA is required. Some species have softer coherent, like such as catfish and zebrafish, so they're easy for microinjection. Small volume of the solution, like 1 to 2 microliters containing more than 10 to 7 copies, should be injected. The rate of survival integration of transgene after microinjection varies widely in different species of fishes and in different batches of the same species. Another method is electroporation. It uses a series of short electrical pulses to make the membrane porous and permeable to DNA incorporation. So a short electric pulse is given, so membrane becomes porous and then DNA can easily enter through those pores. Embryos and sperms can be electroporated. It is less labor intensive and doesn't require special expertise for gene transfer as needed in the case of microinjection. So this is easier than microinjection. It is easier to do this in spermatozoa than in embryos which possess tough chorion. The gene transfer efficiency and integration rate do not differ much between electroporation and microinjection methods. So efficiency wise both are same. In case of zebrafish, 0.1 millisecond pulse of 125 per centimeter for batches of 200x. This is the pulse which is given for introducing gene into fish eggs. So once you introduce the gene in fish eggs, then one has to detect for the transgene whether the gene has been integrated into the host genome. So there are several ways and means to detect this. Most of them use southern blot and northern blot to detect the transgene using a suitable probe. So one can do southern blot, northern blot, or now PCR also can be used for detection of the transgenes. So this one reporter genes are also used. So the reporter gene is defined as a gene whose product detect or mark the cell tissues organism that express the gene from those that do not. So this is another one. Initial stages one can use the reporter genes. So one can easily detect the transgenes. Reporter genes isolated from prokaryotic E. coli are used in fishes like Z gene, cat. Okay. Recently, luciferase and green fluorescent proteins are very commonly used. So they are easy to detect this luciferase and green fluorescent protein. So detection is much easy. So these reporter genes are introduced into fishes and one can easily detect the transgene using this. So glowfish actually is one of the initially developed transgenic fish. So the glowfish is a patented brand of genetically modified fluorescent zebrafish with bright red, green and orange fluorescent color. Okay, so they have introduced reporter gene, particularly the fluorescent protein to create glowfish. So they are ornamental fish actually, and they are now they have been patented. The transgenic zebrafish expressing a red fluorescent protein from a sea anemone under the transcription control promoter from the myosin light peptide 2 gene of zebrafish. So they use a red fluorescent protein. So this protein, red fluorescent protein, was obtained from sea anemone and put under a promoter myosin light peptide 2 gene promoter from which is obtained from zebrafish, and they created the new transgenic zebrafish which is now glowing red in color. So here you can see here red color. Zebrafish have been genetically modified to access fluorescent color. So some of these this is red color from sea anemone or green fluorescent protein, okay, and the orange color. So there are different colors of zebrafish have been colored. So all these are transgenic fish. expressing reporter genes, particularly the fluorescent protein from sea anemone and other sources. So they are ornamental fish. So this is glowfish with expressing green fluorescent protein, electric green tetra. So this is another variety of transgenic. So this is another transgenic zebrafish, which is genetically modified to have long fins, as you can see, long fins here. Okay, so this is modified zebrafish. These fishes are developed mainly for ornamental purpose, to have beautiful colors. And so another important gene of interest, which is very useful in fishes, is growth hormone gene. So that one can have large large size of fishes. So growth is a complex biological process involving genetic, hormonal, nutritional, and environmental factors. So growth hormone is produced by the anterior lobe of the pituitary gland. So the main role of the growth hormone, it increases growth by stimulating appetite and improving the food conservation efficiency. So growth hormone is a protein hormone having a molecular weight of about 22 kilodalton. Transgenic fish carrying growth hormone gene will produce growth hormone endogenously bypassing the necessity of exogenous hormone treatment. So once you introduce the growth hormone gene into fish, so those transgenics will keep continuously produce growth hormone, and then you can have large sizes of the fish. So the growth hormone gene has been cloned in some fishes either from the genomic library or from the cDNA library. So you can obtain growth hormone genes from many sources. So this is a transgenic growth enhanced tilapia. You can see this is the normal tilapia, small in size, but now growth hormone gene has been introduced in these fishes. You can see the large size of these tilapia, which are now you can have large amount of meat from these fishes. So this is another one, transgenic growth enhanced loach. So this is wild type. You can see a small size of loach actually here, wild type. So now the growth hormone gene is introduced into this fish. Now you can see the very large size of these fishes now. So this is the utility of growth hormone. You can have very large size. You can have more uh, meat actually for consumption. So another important gene which can be used for creating transgenic fish is antifreeze protein. So we'll just go in some detail about the antifreeze protein, what is it and how it is useful. So production of cold recent fish variety is useful for establishing aquaculture industry in the in the temperate region where water gets frozen during winter. So particularly in the cold places where the agriculture is not possible, or where there is a lot of snow and all that one. So one can have cold resistant fish varieties. So you can establish aquaculture there for their daily consumption. The gene responsible for imparting cold resistance was cloned from winter flounder, which lives in the polar sea. So it's a variety of fish actually, winter flounder, which can survive in polar sea under extreme temperature. So this species avoid freezing of its blood even at -7 degrees Celsius. If the temperature goes below -7 degrees Celsius, it can avoid freezing, so it can survive and temperature by producing a set of antifreeze proteins. So AFP, these are all short term, it's called AFP, antifreeze protein. So these fishes, this flounder, a winter flounder, which survives in polar sea below -7 degrees Celsius, they produce a set of antifreeze protein which help them in surviving those extreme cold temperatures. This AFP protein are produced in the liver and exported to the bloodstream. So there in the bloodstream, in the blood, it protects the blood uh, from the freezing temperature. So cold temperature, if the temperature water is very low, cold temperature, they pose problem for the fish in water because they are hypoosmotic. So freezing point is higher in their extracellular fluid related to the ambient ambient seawater. So freezing point is higher in their extracellular fluid relative to the ambient seawater. Like in this, it will be easy to understand. Freshwater freezes at 0 degrees Celsius and seawater freezes minus 1.89. But hypoosmotic fish might freeze at minus 0.7. Okay, so because they are hypoosmotic and their freezing temperature is quite high as compared to the seawater here. So at though the temperature of seawater may be minus 1.89 and the fish may freeze at 0.7 degree also. So cold water fish have more NaCl in extracellular fluid, use osmolytes such as glycerol, or they have another mechanism which is called antifreeze protein. So antifreeze protein is 200 times more effective than NaCl. So not freeze until minus 6 degrees Celsius. Even if the ocean temperature reaches below minus 6 degree, they won't freeze because it is more effective than NaCl, sodium chloride. And this is a very useful protein nowadays. It can be used for cryopreservation, food preservation purpose also. So this is the Antarctic fish, Pagothenia, which also which has also make antifreeze protein. So this is the structure of antifreeze protein gene. Antifreeze protein. So this is this is molecular structure antifreeze glycoprotein. Say glycoprotein here. Some are multigenes that have experienced multiple gene duplication and also so there is, you can see disaccharides here, and this is so there are different types of antifreeze protein in 4 different types of antifreeze protein here. One can see they have different structures here. Some is this one is particular type is 129 amino acid. This one is 60 around to 62 to 64 amino acid. So there are different types of antifreeze protein here. This is another structure of antifreeze protein from the cold water ocean pot with the ice binding portion in the lighter blue. So this lighter blue is the which binds the ice now. So this is protein of structure of the antifreeze protein, and the lighter one is the one which binds through the ice binding portion of the antifreeze protein. So there, as I as I mentioned in the earlier slide, so there are different types of antifreeze protein. So this is from the ocean pot. This from the winter flounder, another fish, winter flounder from the Arctic pot. And there are some different organisms also besides fishes. Some insects also, they also can survive extreme cold conditions. So they also make these antifreeze protein. Like this is from the yellow mealworm beetle, and this is from budworm moth, snowflea here actually. And the lighter portion in all these is the one which binds the ice now. Okay. So there are different types of antifreeze protein and they have different structure also. But the common thing in all these is that they have a domain which binds to the ice. This is called ice binding domain. So how does it actually work? So antifreeze protein lower the temperature at which ice crystals grow by attaching to existing ice crystal and avoid tissue damage. So what do they do? These antifreeze protein, they bind to the ice ice and then prevents its further growth by, okay, and then help in avoiding the tissue damage. Suppose if the ice crystal keep on growing, they can damage the tissue and the fish may die. So the AFP then protects them, um, prevent the freezing of these fish. So basically they lower the freezing point and do not upset the osmotic balance of the fish. Without upsetting the osmotic balance of the fish, they lower the freezing point. So even the temperature goes below zero, fishes can survive. So the hydroxyl amino group of the AFP, hydroxyl amino group of the AFP are assumed to form hydrogen bond with the oxygen atom in the ice. Oxygen atom in the ice. So this is how they form the bond. They form the bond with the ice molecule. AFP are adsorbed to the ice crystals and growth on the prism faces is stopped. So once the antifreeze protein binds to the ice crystals, which are being formed in the fish's blood, okay, and then it prevents the further growth of those ice. Okay, so the further ice formation is not does not occur once the AFP antifreeze protein binds to the ice crystal. Their further growth will be prevented, and hence fish will be able to survive. So this stop the growth on the basal plane of the crystal. Ice cannot spread over an FG molecule, so it will grow in between the molecules. So basically, it will prevent the further ice formation, and then fish will be able to survive in extreme low temperature condition. So basically, AFP then stop the growth of the crystals, and further ice formation will be prevented. So this picture will be clear actually. One can see ice crystal growth pattern, the presence absent of FP. So this is what they have done. They have taken the antifreeze protein and did some assays actually, how actually it prevents. So here one can see here. So this is the ice crystal here in absence of antifreeze protein. Here one can see a large crystal, ice crystal here, ice crystal. This is ice crystal here, large ice crystal in absence of AFP protein. When the crystal grows in, and when the AFP antifreeze protein is added, the crystal size is doesn't increases further. And here when they use large amount of antifreeze protein, the crystal growth was further prevented. So you can see very small size of the crystal. So this is how the antifreeze protein helps the fish survive in the extreme cold temperatures in Arctic or Antarctica. So this is how the antifreeze protein works. They bind to the ice and prevent it further growth. So this is this is one of the important protein, antifreeze protein, which can be introduced now into the other fishes which grow in warm regions like salmon and all that one. So these antifreeze protein gene can be introduced in salmon, and then these proteins can be easily cultured in cold climate condition like Arctic or Antarctica, and which or very other cold places where actually other cold conditions where this can be easily grown now with the help of antifreeze protein gene. So another important trait which can be introduced in transgenic fish is disease resistance gene. So fish has poorly developed antibody dependent immunity. So they basically lack antibody dependent immunity like we humans have got that one. Effort to produce disease resistance in fish stock by transgenic has begun recently. The potential rainbow trout lysozyme gene as a bacterial inhibitor was assessed in the Atlantic salmon. So development of vaccine for the fishes. Fish vaccine will greatly aid the fish industry, but very few are available. Fish are not easily vaccinated. It is usually done by injection or by immersing them in water with vaccine. So you can put vaccine in the water, and then it will be taken up by the fish, and then they can get vaccinated. Using ultrasound can facilitate vaccine entry into the fish. Vaccine is produced by isolating and expressing gene encoding viral proteins. Okay. So these are some of the examples which vaccine has been produced. The first, there's some example of transgenic fish now. We'll go through some examples. The first transgenic fish were produced in China in 1985. Okay, so this first example. As of 2013, approximately 50 species of fish have been subject to genetic modification. This has been resulted in more than 400 fish traits combination. Most of the modification have been conducted on food species such as Atlantic salmon. So this is one of the commonly consumed fish, tilapia, and common carp. So these are some of the example commonly consumed fishes which have been made transgenic. In 2015, the Aqua Advantage salmon was approved by the US Food and Drug Administration. So this already transgenic fish has been approved by FDA for commercial production, sales and consumption, making it the first genetically modified animal to be approved for human consumption. Some GM fish that have been created have promoters driving an overproduction of all fish growth hormone. This is a dramatic growth enhancement in several species, including salmon, carps, and tilapias. So this is the acquired advantage salmon. In November 2015, the FDA of the US approved the GM, that is genetically modified acquired advantage salmon created by Aqua Bounty for commercial production, sale and consumption. It is the first genetically modified animal to be approved for human consumption. So this is the fish is essentially an Atlantic salmon with a single gene complex inserted, a growth hormone regulating gene from a Chinook salmon with a promoter sequence from an ocean pot. So this permits the GM salmon to produce growth hormone year around rather than pausing for part of the year as do wild type Atlantic salmon. So now in this particular aqua advantage salmon or a transgenic salmon, growth hormone gene is produced throughout the year. So that's because of that it keeps on growing in size. So the wild type salmon takes around 24 to 30 months to reach market size, that is 4 to 6 kg. Whereas the producer of the GM salmon claims it requires only 18 months for the fish to achieve this one. So now the size of so it grows very fast. It takes less time to gain a large amount of weight as compared to the wild type fish. Aqua Bounty argued that their GM salmon can be grown near to end markets with greater efficiency. They require 25% less feed to achieve market weight than the Atlantic salmon, which are currently reared in the remote coastal fish farms. They are making better for the environment with recycled waste and lower transport cost. It has been claimed that to prevent the genetically modified fish breeding with the wild salmon, all fish will be females and reproductively sterile. So their current and potential application of transgenic fish, growth enhancement is the major one. So most of the companies are looking for growth enhancement using growth hormone gene. Freeze resistant using and cold tolerance. So this is using antifreeze protein. So the common fish like carp, salmon. So these genes, antifreeze genes, can be introduced in common carp, salmon, and then they can be grown in extreme cold temperature like Arctic or Antarctica. So that aquaculture can be done in those places under extreme cold condition. Salinity tolerance, if the salt concentration is very high, then these fishes can be made tolerant to high salt concentration. Another trait is disease resistance, so that aquaculture particularly is important in aquaculture. One can have disease resistance. Metabolic modification, improved product for the consumer, fish farming, production of pharmacology proteins. Some important pharmaceuticals can be made in fishes because introducing genes in fishes is easy as compared to other animals. So this pharmaceutical can be produced and then obtained in large amounts. So transgenic fish are used in research covering five broad areas: enhancing the trait of commercially available fish, particularly the growth enhancement. They're used as bioreactor for the development of medically important proteins. Okay. Some like bioreactor, particularly they can be used for making large amount of therapeutic proteins for treatment of various diseases in human. So some of these fishes can be transgenic fishes can be used an indicator of aquatic pollutants. So this is very important for our environment purpose, for environmental purpose. Also, these fishes can be used. Developing new non-mammalian animal models, particularly zebrafish, can be used for animal model for basic, several studies like toxicity studies, safety studies, and functional genomic studies also can be easily done in with the help of these fishes. So these transit fish can also play a very important role in research R&D. So most GM fishes are used in basic research in genetics and development. Two species of fish like zebrafish are most commonly modified because they have optically clear chorions. Cells develop rapidly. The one cell embryo is easy to see and microinjected with transgenic DNA. Zebrafish have the capability of regenerating their organ tissues also, so they are so they're very useful for these studies. They are also used in drug discovery also. GM are being explored for benefits of unlocking human organ tissue diseases and failure mysteries. For instance, zebrafish are used to understand heart tissue repair and regeneration in efforts to study and discover cures for cardiovascular diseases. So their zebrafish is now playing very important role in this in pharmaceutical research also. Transgenic rainbow trout have been developed to study muscles development. The introduced transgene causes green fluorescence to appear in fast-twitch muscle fibers early in development, which persists throughout life. It has been suggested the fish might be used as an indicator of aquatic pollutant or other factor which influence development. Okay, so so using this type of transgenic fishes like rainbow trout, they can be used as an indicator of aquatic pollutants, so which basically influence the development of these fishes. In intensive fish farming, the fish are kept at high stocking densities. This means they suffer from frequent transmission of contagious diseases, a problem which is being addressed by GM research. Grass carp have been modified with a transencoding for human lactoferrin, which doubles their survival rate related to the control fish after exposure to Aeromonas bacteria and grass carp hemorrhage virus. So lactoferrin actually has this is human lactoferrin which, present in human milk and all that one. So this has antibacterial properties. So this particular gene, human lactoferrin gene, can be introduced into the carp, so they can become resistant to these infection from Aeromonas and other bacteria. So this is another. So they one can have disease resistant carp using lactoferrin, human lactoferrin. Similarly, cyclopin has been used in channeled catfish to enhance their protection against several pathogenic bacteria by 2 to 4 times. So basically, these genes can be lactoferrin, cyclopin. They can be used basically to have disease-resistant transgenic fish for aquaculture purpose. So there is another potential of transgenic fish to detect aquatic pollution. Several research groups have been developing genetically modified zebrafish to detect aquatic pollutant. The lab that developed the glowfish originally intended them to change colors in the presence of pollutant as environmental sentinels. So basically, they change colors in the presence of pollutant. So that will give the indicator that the pollutants are there in the water. So this very easy way of detecting aquatic pollution. Teams at University of Cincinnati and Tulane have been developing GM for the same purpose. Several transgenic methods have been used to introduce target gene in zebrafish for environmental monitoring, including microinjection, electroporation. As I told earlier, microinjection is the most commonly used method to produce transgenic zebrafish, as this produces the highest survival rate. So, as food safety of transgenic fish, so this is an important issue. Like more, several organizations, they are concerned about the safety of these transgenic products. So the GM food safety depends on the nature of the gene, the transient product it encodes, and the resulting phenotype. Ethics and the animal production concern allow the development of healthy and safe fish only. Transgenic fish have received extra copies of growth hormone gene, resulting only moderately raised levels of circulating growth hormone. Growth hormone is a protein hormone which is degraded along with all other food proteins. So basically, it's these transgenic fish is safe to consume because the gene introduces a growth hormone gene, and this growth hormone gene is a protein and which can easily get degraded when it is cooked or when it is consumed by the human. So this gets degraded very fast, so it won't be able to create much problem in the human body because it get degraded very fast in the after cooking and human consumption. So food safety issue won't be much as with help in case of transgenic fish for consumption. So meat from fish modified with growth hormone is regarded as completely safe for human consumption. Concern have been voiced of possible risk of consumption of transgenes, their resulting protein, potential production toxin by aquatic transgenic organism, changes in the nutrition composition. So these are some of the concerns which can be taken care of. The one important aspect is that the immune response of the transgenic products, like allergic reactions and other things due to the transgenic genes and products. So this may be one of the concern which needs to be taken care of when we think of transgenic food for consumption. Other concern is the environmental impact of this transgenic fish. The possible impact from escape of genetically modified organism from aquaculture facility are of great concern to some scientists and environmental groups. Critics and scientists predict that GM fish could breed with wild population of the same species and potentially spread undesirable genes. Escaped transgenic fish could harm wild fish through increased competition or predation. The consequences of such competition would depend on many factors, including the size of the wild population, the number and specific genetic strain of the escaped fish, and local environmental conditions. So these are some of the concern with regards to the transgenic fish, which needs to be addressed. FDA could require that only sterile GM fish be approved for the culture in ocean pens. So now what they have done, FDA has said that only the GM genetically modified fish should be sterile so that it cannot compete with the wild varieties. Fertilized fish eggs that are subjected to heat or pressure shocks retain an extra set of chromosome. So this is basically it will make so they will have extra set of chromosomes, so they will make triploid. So once the fertilized fish eggs are subjected to heat or pressure shock, they can retain an extra set of chromosome. So instead of diploid, they will become triploid. So the resulting triploid fish do not produce normal eggs or sperms, and so basically they are sterile. And females do not exhibit maturation of the ovary or reproductive behavior. So this is basically if you have a triploid fish, they will be sterile, and they won't be able to then compete with the wild one. So thus all female lines of triplet fish are the best current method to ensure non-breeding population of genetic modifiers. So this is one of the way we can address the environmental impact of the transgenic fish, just to have a sterile genetically modified fish by making them triploid. Triploid fish will be sterile. So there will be what are the possible benefits and disadvantages of GM fish and seafood. Biotechnical proponents maintain that genetic modification has many advantages over traditional breeding method, including faster and more specific improvement of beneficial traits like growth hormone, antifreeze protein, disease resistance. So this will all help in uh, making aquaculture much easier. So genetic modification allows scientists to precisely select trait for alteration, enabling them to create an organism that, for example, grows larger or faster, has a different nutritional content. Increased freeze resistance in fish could lead to the ability to grow freeze-resistant species in previously inhospitable environment, allowing aquaculture to expand into previously unsuitable areas. So the antifreeze protein also have a lot of basically advantage. One can grow these freeze-resistant genetically modified fishes in extreme cold conditions. So biotechnology proponent they claims these advantage could translate into a number of potential benefits such as reduced cost to producer. So it will be more economically viable for the farmers, lower prices for consumer for edible fish and pharmaceuticals, and environmental benefits such as reduced water pollution from the waste. Majority consumers in the industry appear to have accepted GM food and feed crop. Basically, in United States, so they have already started consuming this transgenic fish with human with the growth hormone gene. So it already being consumed. So there is no safety till now. There is no safety issues. Just to conclude, the future success and application of transgenic fish will depend upon by a successful demonstration of a lack or potential lack of environmental risks, food safety. But if food safety is important, it shouldn't cause any allergic reaction or higher immune response in the human body. Appropriate government regulation and labeling, public education is also very important. So we need to educate public that these transgenic fish are safe for consumption, and development of genetic sterilization for transgenic fish. Like one can have triploid fishes so that they are sterile and they don't mix with the wild varieties in the ocean. Appropriate well executed public education may be necessary to gain broader consumer acceptance of the transgenic fish from an environmental standpoint and perhaps in relation to how organic a transgenic fish may be.